Okay, I now invite you to the adaptive immune system. So, the adaptive immune system, also called the acquired immune system, happens to be the second line of defense. It's the second line after the innate immune system. So, unlike the innate immune system, which we saw that is non-specific and acts immediately, this adaptive immune system is highly specific, but it takes time to develop. And after it has developed, it provides immunity for a longer time. So we can say it provides long-term immunity. So adaptive immunity, you are not born with it. It's something you acquire. It's something you get possibly as a result of an adaptation to something. So that's why it's adaptive or acquired. So what are the types of acquired immune system or the acquired immunity, which is also called the adaptive immunity? So acquired immunity or adaptive immunity refers to the immunity that an individual develops after exposure to a pathogen or through the stimulation of the immune system. So you have this acquired immunity after you have an infection, after you are infected na ugonjwa fulani. Ukipona, then you will have acquired that immunity. Or you can have this acquired immunity when your immune system has been stimulated. How does your immune system get stimulated? Through vaccination. We will see that towards the end, but the stimulation of your immune system inatendeka through vaccination. So the classification of the acquired immunity is based on how the immunity is gained, how you developed that immunity. So the first type is the active immunity. That is the first type of acquired immunity. So active immunity occurs when the immune system is stimulated to produce its own antibodies and memory cells in response to an infection or vaccination. So either you are vaccinated or you are infected. That is active immunity. So that your immune system produces its own antibodies. So usually, the active immunity as a type of acquired immunity provides long-lasting protection. So an example for is if you are vaccinated against flu, that vaccine will trigger your immune system to produce antibodies against the flu, that particular flu. So in the future, in case you are exposed to an infection, a flu infection, the antibodies that your immune system was triggered to produce will prevent or will protect you against that infection. So that is the active immunity. The second is the passive immunity. So passive immunity occurs when a person receives ready-made antibodies from another source. Okay? So you receive the antibodies that have been made at a different source. It is the antibodies are not being produced by your own immune system, unlike what was happening in the active immunity. The passive ones, unapatiwa ile iko tayari. So this immunity is immediate because you get the antibodies zikikuja, kumaanisha ziko tayari kufanya hiyo kazi. So the immunity is, the response is immediate, but it is short-lived. Why is it short-lived? There are no memory cells that are formed by your own immune system. umepatiwa the antibodies from a different source. So it means the memory cells are where you got the antibodies from. So the example is if a newborn baby gets antibodies from the mother, either through breastfeeding or through the placenta. So with such, the newborn baby will, through breastfeeding, will find temporary protection because the antibodies came from the mother. They were already prepared by the mother's immune system. Ndio mtoto akazipata tu through breastfeeding. So that is passive immunity. Primary immunity happens when it is the body's first response to a new pathogen. So it means your body has never been infected by that pathogen before. So primary immunity is the first response by your body to that particular pathogen, which is new. So because the pathogen is new, then the primary immunity, its response will be slow. Because your immune system needs time to recognize that pathogen before it can produce antibodies that are specific to it. Ni kama unaona kitu yenye unafikiria it is dangerous, but you don't know what it is. So you are not sure what will handle that thing. Kama ni fimbo, kama ni panga, kama ni kupiga kelele, you are not very sure. So by the time you figure out that let me use a panga, it will have taken time. So the primary immune system, the primary immunity as a type of acquired immunity happens when it is the first time that your body is responding to that particular pathogen. The pathogen is new. So your immune system hasn't recognized that antigen, that pathogen. So it does not know which antibodies to produce. So it will take a bit of time. That's why the primary immunity is slower. Okay. So for example, if you are exposed to chickenpox for the first time, your body will take a few days or weeks before it can mount an immune response. Why did it take a few days or a few weeks? Because it was trying to recognize that chickenpox causing pathogen, and once it has recognized it, it will now be able to mount an immune response that is specific to chickenpox. So the antibodies that it generates are the ones that are specific to chickenpox, are the ones that will now be able to fight off the chickenpox infection. So what about secondary immunity? So secondary immunity is faster. It is stronger. Why? Because it happens when your body or the body is now encountering a pathogen that it has previously fought. So secondary immunity occurs after the primary immunity occurred before. So it is faster, it is stronger, because your body is able to remember. Okay? So there are memory cells that were created during the primary response. So those memory cells were kept. So now that there is the same pathogen which is coming now to attack your body can now trigger an immune response, ikiangalia inaona hapana, hii nimewahi kupambana nayo na nilitumia hii. So it will go and now bring those memory cells. So because the memory cells were already formed, that will make the immune system to be, the time of reaction to be faster. Okay. So if you had, for example, been infected with chickenpox before, your immune system will remember that virus. Okay. And anytime you now have that chickenpox virus attacking you again, your immune system will remember it and it will quickly destroy it before the symptoms appear, even if you are exposed to it. Okay. So Specific immunity, which is also called adaptive immunity, is part of the immune system that provides a targeted defense against specific pathogens. It is targeted, it is specific, because it recognizes and remembers particular antigens. So if it remembers that particular antigen, that particular pathogen, then a specific immunity will be stronger, will be faster, wherever there is a future encounter. Sababu inakumbuka, ina recognize na inakumbuka. Anytime you see a snake, something that you know is dangerous, if you saw a lion and a gazelle, you will begin running away from the lion. But if you saw a wild animal yenye inakula watu na hauijui, you will not be sure should you run away or not. But once you can recognize that this is a dangerous animal, you will have a stronger and a faster response. Stronger because you know, this is dangerous. The decision-making process is fast. Then after you've made that decision, you now respond equally faster by running away. So the specific immunity or the adaptive immunity involves B cells and T cells. These ones are the ones which will now generate long-term protection through the memory cells. So B cells and T cells ndio zinaleta long-term protection. Hizo ndio zitatumika as the memory cells. So specific immunity or the adaptive immunity originates from specialized white blood cells, what we call the lymphocytes. These lymphocytes are produced and matured in primary lymphoid organs. You can check in the lymphatic system when you read your notes or you listen to the video about the lymphatic system, you will see the primary lymphoid organs. Okay. So the primary lymphoid organs where the lymphocytes are produced and mature there happen to be these two. One is the bone marrow. So the bone marrow is where the B cells develop and mature. So bone B. So bone B. In the bone marrow, B cells develop there. They mature there. And these B cells, kazi yake, they produce antibodies. Antibodies are produced by B cells. The function or the work of these antibodies is to neutralize the pathogens. The other lymphoid, the primary lymphoid organ, the other primary lymphoid organ is the thymus. So we saw that from the word bone marrow, ukiona B ya bone, hiyo ndio B cells sasa. So in the bone marrow, B cells develop there, B cells mature there. But in the thymus, that is where the T cells, so from the word thymus, kuna T, then we can also remember that T cells. So T cells mature in the thymus. The function of the T cells is that they recognize and destroy cells that are infected or cells that are abnormal. Infected cells are the ones that will have a pathogen inside them. But abnormal cells are cells that could be possibly cancerous. Zikuwe na yani zinafanya structure yake inakuwa abnormal. Zile proteins zinatengeneza are not normal proteins. Okay? So the T cells will recognize and destroy cells that are infected and cells that are abnormal. So once the T cells and the B cells are mature, they will be circulated through the bloodstream and through the lymphatic system. As they are being circulated around the body using the bloodstream and the lymphatic system, kazi ya sasa hii B cell na T cell ni kuscan kuangalia kama kuna pathogens. Okay? Kuangalia kama kuna pathogen. So when they encounter an antigen, ile imetengenezwa na pathogen, they will activate and mount an immune response. So B cell, T cell zinazungushwa na bloodstream na lymphatic system around our body. Kazi yake ni kama zinafanya patrol. Once they encounter an antigen is something that can stimulate an immune response. Okay? So once they encounter an antigen, the B and T cells will activate and mount an immune response. So once they've activated and they've mounted an immune response, they will now create memory cells. The memory cells, hizo ndio zitasaidia sasa in future for a secondary response, kama kutakuwa na ingine. Okay? So the memory cells that are created ndio zitakumbuka that pathogen, ndio zitakumbuka that antigen. So these memory cells, kazi yake, they will ensure that there is a rapid and effective reaction in the future when there is an exposure to the same pathogen. So if you had chickenpox, there will be memory cells for that virus that causes chickenpox. So when you are exposed to the same virus again at some point, these memory cells will bring forth a rapid and effective reaction because the pathogen is the same. So that is a secondary immunity. So the example here is after receiving a measles vaccine, your immune system will produce B cells and T cells that will remember the measles virus. So in the event the measles virus enters your body, those memory cells will quickly eliminate that measles virus before that virus can cause illness. Okay. So there was an infection, there was an exposure earlier to a vaccine for measles. That vaccine for measles led to the creation of antibodies. Okay. Led to the creation of antibodies against the measles virus. So when the measles virus eventually now enters the human body, which already had the measles vaccine, the antibodies, the B and T cells, zenye zimetengeneza now the memory cells, those memory cells that are specific to the measles virus, ndio zitakuwa deployed haraka so that zikuje eliminate hiyo measles virus even before a disease or an illness is caused. So what are the key characteristics of adaptive immunity? So as we've seen, adaptive immunity is a specialized defense system which helps to protect the body from infections by recognizing and responding to specific pathogens. So it is specialized. It recognizes and responds to specific pathogens. So let's look at some of the characteristics. So the first one is specificity. So their adaptive immunity is highly specific. That means immune cells will target particular pathogens, okay, based on unique molecular structures that are called antigens. So a pathogen is what will elicit an immune response. But a pathogen will be produced by a particular pathogen. An antigen will be produced by a particular pathogen. Okay? So these antigens are found on the surface of invaders. So a pathogen will have an antigen on the surface of that pathogen. Okay? So it means a bacteria will have a different antigen and a virus will have a different antigen. So your immune system will respond to the antigen of a bacteria differently as it will respond to the antigen of a virus differently. Okay. So if you catch flu, for example, your immune system will generate antibodies that are specific to that particular type of flu. Okay. So if you have a new strain that will appear, your immune system may not immediately recognize it because ile yenye ulikuwa exposed kwake mapema ni tofauti na hii yenye imetokelezea. So the memory bit is not there because your immune system is specific. So it can only respond to the same type of flu, but it cannot respond to a different one. The second component or characteristic is memory. So a major difference between adaptive immunity and innate immunity is the ability of the adaptive or the acquired immunity to remember pathogens. So you only remember what you have already encountered. Ukiambia mtu anakukumbuka inamaanisha ulimuona mahali. Okay? Ndio utasema anakukumbuka. So the same thing happens with the adaptive immunity. Itakumbuka ile yenye imewahi kukutana nayo, what is it has encountered before. So after an infection has happened, immune cells will store information about that pathogen. So the memory cells. Okay. So when the same pathogen again tries to infect you, there will be a faster and stronger response because there was information that was stored about that pathogen. Okay. The third characteristic is clonal expansion. So when an immune cell recognizes an antigen, that immune cell multiplies rapidly so that itengeneze many copies of itself. Okay. So that expansion, it creates more identical cells that we call clones. So that clonal expansion, it's expanding. Inataka kupata wale wenye wataisaidia wengi. So it photocopies itself so that it can create many copies of itself. Okay. So these identical clones will now be equipped to fight off that particular infection effectively. So after the memory, there is an expansion, there is an increase, there is a photocopy, so that the immune cell yenye ni moja imegundua kwamba there is a foreign invader. But hiyo cell peke yake haiwezi ikadeal na hiyo invader. So lazima itengeneze many copies of itself. It has to expand the numbers. Okay, so that's what we are calling the clonal expansion. It has to multiply rapidly. Okay, so that is the clonal expansion. Because peke yake haiwezi to deal with that antigen. It needs to mobilize so that zikuwe mingi. That expansion, that increase, because it's an increase of itself, it creates many copies of itself. Okay. So that creation of identical copies is what we call cloning. Okay. So this is the clonal expansion. The fourth characteristic is self-tolerance. So there has to be a way in which the immune system can now recognize what is foreign so that it can attack it and avoid to attack what belongs to the body. So what helps this component is what we call the process that we call self-tolerance. So that the immune cells are taken through some certain sort of training so that they know how they can ignore the proteins of the body and how they can attack the proteins of a cell that does not belong to the body. Okay. So in the event that this process of self-tolerance fails, sasa hapo tunakuwa na what we call the autoimmune diseases. When now your immune system begins to attack your own body cells. So the immune system if it's working optimally, it is supposed to recognize and tolerate the cells za mwili wako. Ni kama ni kwa mpira, you should know who is your teammate and who is an opponent. Because when you can identify who is your teammate, you will know whom to pass the ball to. But when you've identified or recognized who is the opponent, you know whom to mark. Okay, so that's the whole concept of self-tolerance. In a football match, in any match, teamwork, you must be able to recognize whom are you supposed to mark and whom are you supposed to pass the ball to. Okay, if you pass the ball to someone who is not an opponent, they will not score in your own goal. But if you pass the ball to an opponent, they will score. Another component is diversity. So the immune system deals with different antigens. Okay, it encounters different antigens. So it should also be have an ability to recognize the many pathogens zenye inakutana nayo. Okay, so it should recognize the different antigens, and what makes it to be able to recognize those different antigens is the structure of the antibody. We'll look at that. We'll look at the structure of the antibody next. Okay. So the ability of the immune system to recognize different antigens, that is important, so that your immune system should be able to recognize if you have a bacterial infection, if you have a viral infection, if you have a parasitic infection. Okay, so it needs to adapt to the different types of pathogens that it will encounter. So that's the whole concept of diversity. So the other characteristic is the slower initial response, but long-lasting protection. This is the adaptive immunity you were not born with. So initially, when you have the primary exposure, your immune system will not know how to respond. But once it has encountered it, it will keep memory and have that long-lasting response. Okay. So the adaptive immunity takes time because it has never been exposed to that pathogen. But once it has been exposed to it, it will have now memory cells, B and T cells, which are memory cells. So anytime it is exposed to it, there will be an aspect of memory and it will respond fast. So the aspect of slow initial response and long-term protection is a principle that vaccines rely on. Okay? So the first dose may take weeks to develop immunity, but booster doses strengthen the response and ensure that there is a long-term or long-lasting protection. So once you are vaccinated or immunized, that vaccine will trigger your immune system to produce B and T cells, the antibodies. But that production will take time. But once they are produced, they will now have memory cells that are kept within the immune system. So wherever there is something that happens, then the immune response will be fast. Okay? The immune response will be fast. And because of these memory cells, they will now have a lasting protection against what you've been vaccinated to. Okay? So what are the components? What is it that now makes the adaptive immune system to function? So the adaptive immune system relies on lymphocytes. Lymphocytes are white blood cells. These white blood cells, these lymphocytes, they have the ability to recognize and respond to specific pathogens. Okay, so the components of the adaptive immune system, what we are seeing is that the adaptive immune system relies heavily on lymphocytes. What these lymphocytes do, they have to recognize, and after they have recognized, they have to respond. It's not enough for you to know that there's a thief in the house. What did you do after recognizing that there was a thief in the house? You have to respond. Okay, so that's what these lymphocytes are doing. So there are two main types of lymphocytes that play a major role in the adaptive immune system. So the first is the B cell. So the B cells are critical for the humoral immunity. So the B cells are produced in the bone marrow and they remain there, so they mature within the bone marrow. The main responsibility of these B cells is that they produce antibodies. So antibody production is a function of the B cells. So each B cell has a unique receptor that makes it to bind to a specific antigen. So there will be a B cell that will, for example, bind to a bacteria. There will be a specific B cell that will bind to a virus. There will be a specific B cell that will bind to a parasite. So once the B cells have been activated, they become plasma cells. Then now those plasma cells are the ones that will now produce antibodies. So you have a B cell which is not activated, but once it is activated, it becomes a plasma cell. Then that plasma cell now is what will produce the antibodies. But not all B cells will change to plasma cells because we need to have some memory B cells. So some B cells will remain to become memory B cells. Those memory B cells ndio zitasaidia for a faster response in case the same pathogen returns. Okay, in case the same pathogen returns. So the key players in this case, one are the plasma cells. These are the ones that are created after activation of a B cell. And once these plasma cells have been formed, their main function is to produce antibodies. The other bit are the memory B cells. The function of the memory B cells is for long-term immunity. Hizo ndio zitakaa hapo zikikumbuka for memory. So that whenever there is the same pathogen attacking, then these memory B cells can be mobilized fast so that they can attack that pathogen. The second component of the adaptive immune system are the T cells. So the B cell was responsible for the production of antibodies. Antibodies lead to the humoral immunity. But T cells, we looked at it in the T cells and cell-mediated immunity. So T cells are responsible for cell-mediated immunity. Okay. So they are also produced in the bone marrow, but they mature in the thymus. What they do is they don't produce antibodies, but they help in the elimination of both infected cells and abnormal cells. So the T cells do not rely on antibodies to function, but still they also help eliminate cells that are infected and cells that are abnormal. So T cells recognized infected cells using a receptor, which is now found on the T cell, the T cell receptors. There are two main types of T cells. I hope you remember that. The helper T cells or the CD4 T cells. The CD4 T cells or the helper T cells, these ones act as the generals of the immune system. So they coordinate, okay? They coordinate the immune responses. So the helper T cells or the CD4 T cells, they release chemicals, the cytokines. So the chemicals that they release are the ones that will activate the B cells, the macrophages, and the killer T cells. So the helper T cells don't kill, they don't engage in the killing themselves. They only coordinate the immune response. They coordinate the immune response by releasing cytokines. The cytokines are the ones that will now activate the cells that will be engaged in the real act of killing. The second type of the T cell is the cytotoxic T cell or the CD8. So these ones act as the soldiers. They kill directly any cell that is infected or abnormal, which makes that cell. The cell could be abnormal as a result of being cancerous. So the cytotoxic T cells kill using perforins. Perforins are the ones that make perforations, iyo mashimo, on the membrane of the infected cell. The second mechanism in which the cytotoxic T cells kill is the use of the granzymes. The granzymes, there's in a later year, programmed cell death, what we call apoptosis. Okay? So the cytotoxic T cells will target cells that are infected with a virus or tumor cells. A tumor cell is cancerous. That cell will be abnormal in a way. Okay. Then the third cell are the memory T cells. The importance of these memory T cells is in the provision of the long-term immunity. By providing that long-term immunity, they help in remembering the past infections so that they can lead to a faster response. So this long-term immunity can persist for years or at times even for a lifetime. There are some vaccines, kama measles, you are vaccinated once. So which means the protection is for a lifetime. But there are others which you need to go in for a booster. So which means the protection has persisted. The memory has persisted just for a number of years. Then you have now to go in for a booster. So how exactly does the adaptive immune system work? So the first thing before the adaptive immune system sets into action, it has to recognize the antigen. So the step one is antigen recognition. So pathogens have unique molecules which are called antigens. An antigen is a molecule that will elicit an immune response. And an antigen is specific for a particular pathogen. So there are specialized cells, which we call the antigen-presenting cells. The examples are dendritic cells and macrophages. These are the ones that will capture antigens and then present them to T cells using the MHC. We saw that. So antigen recognition. The antigen is specific, is unique for a particular pathogen. Then that antigen is picked, is captured by the antigen presenting cells. The antigen presenting cells then present that antigen to T cells. The T cells are now being presented to these antigens by the major histocompatibility complex. So the APC picks the antigen. Then using a major histocompatibility complex, that antigen presenting cell presents the antigen to the T cell. Step two is the activation of the lymphocytes. So the helper T cells will activate B cells and cytotoxic T cells using chemical signals. The helper T cells, those are the generals. They release cytokines, which will now lead to the activation of the B cells or the cytotoxic T cells. Then the B cells and cytotoxic T cells will now multiply rapidly. The clonal expansion, okay? That clonal expansion. So the lymphocyte activation has been done by use of the helper T cells, the generals. They don't engage directly, but they release chemicals. They release cytokines, which go to activate what is it that will now fight, whatever it is that will now kill the infection, the pathogen. After the lymphocytes have been activated, now they can attack and eliminate the pathogen. So B cells will now produce antibodies. The antibodies will bind to the pathogens. Once the antibodies have bound to pathogens, they have now marked them for destruction. Okay? So B cells, kazi yake, they produce antibodies. The antibodies will go and bind to the pathogens. That makes it easier now for the macrophages to come and now destroy whichever pathogen that has an antibody bound to it. Another mode of attack and elimination is through the cytotoxic T cells. These ones will have been activated. So kazi yake, they will directly kill the infected T cells, the infected cells, sorry. Then there are some cells, kazi yake ni to suppress the immune response. So those are the regulatory T cells. Because if the immune response is not suppressed, it can likely lead to autoimmunity. It can destroy other body cells. Then the last and important step is now in the formation of memory. Okay. So after the infection is cleared, there are some B cells and T cells that will remain in the body. Those B cells and T cells form the memory of the body. So immunologic memory inatengenezwa namna hiyo. So in case the same pathogen reappears, the memory B cells and T cells will provide a faster and stronger response. Faster sababu zimeshawekwa, zilitengenezwa kitambo. Okay? So they don't need to be produced again. So because they are there, it's just a bit of clonal expansion. Think of it if you have notes to read and someone else doesn't have the notes. Ule mwenye ako nazo alisha download akasave, ako na uwezo wa kusoma haraka kuliko huyo mwenye anaanza kutafuta kuuliza ziko wapi ndio asome. So antibodies, which are also called immunoglobulins, are Y-shaped proteins. As we've seen, antibodies are produced by B cells so that they can neutralize pathogens. Okay? So antibodies, kazi yake is to neutralize pathogens. And they are produced by B cells. Okay? So the types of antibodies, you have antibodies, we said they're also called immunoglobulins. So you can talk of immunoglobulin G, that is just antibody G. So it's the most abundant and it provides long-term immunity. So it's part of the memory cells. Immunoglobulin A is found in the mucosal areas, and so that is saliva, tears. Immunoglobulin M is the first antibody that is produced in most infections. Immunoglobulin E for allergic reaction. So if you are allergic to something, that is just the function of your immunoglobulin E. Then immunoglobulin D, it helps to initiate the activation of B cells. Remember, the B cells need to be activated so that they can form plasma cells. Then the plasma cells are the ones that will now form antibodies. So how do these antibodies work? How do these antibodies work? Their main function is to defend the body against harmful pathogens, be they bacteria, be they viruses. So how do they do this prevention of the harmful bacteria, harmful pathogens against the body? They do that through three main mechanisms. One is neutralization. The antibodies neutralize by blocking the harmful effects of the pathogen. To neutralize is to block. To whatever was meant to be harmful, if it is blocked, then you will not feel the harmful effects. For people who play volleyball, wanasema you block, mtu akienda strike, you block, okay, so that the ball doesn't come into your court. That is blocking. So neutralization is when antibodies directly bind to a pathogen, preventing it from causing harm. So an antibody will directly bind to a pathogen. Once the antibody has bound to the pathogen, that pathogen might not cause harm. The other way that the antibodies work is opsonization. We saw that, where now the opsonization involves the antibody marking the pathogen for destruction. So through opsonization, an antibody places a mark on a pathogen which tells the macrophage to eat that pathogen. Okay. So when the macrophages come, it's easier for them to know which cell should be eaten because it has already been tagged or been marked for destruction. The third way is the complement activation. So through the complement activation, it triggers a destructive cascade. So the complement system is a group of proteins within the blood that help to destroy the pathogen. So once the antibodies have bound to a pathogen, they can activate this complement activation system, leading to either one, formation of pores. Okay, so kuweka, perforation. So the complement protein can create holes in the pathogen membrane so that that pathogen can then burst because wameweka mashimo on the membrane. The second thing, that it can lead to inflammation. So inflammation, it will lead to the attraction of more immune cells to that site of infection. Remember, inflammation, the most important thing is histamine. Histamine increases, vasodilates the vessels. Upon vasodilation, then blood will flow to that area because of a reduced pressure. The third thing that will happen is the enhanced phagocytosis. Okay. So some complement proteins will help the immune cells to engulf and remove the pathogen more efficiently. So there is the formation of pores, kuweka mashimo kwa membrane of the pathogen. The second thing is inflammation, where more immune cells are brought to this infection site. Then the third is enhanced phagocytosis, where now some complement proteins make the immune cells more able to engulf, and after they've engulfed the pathogen, they can now destroy or remove it easily. Okay. So together, these three mechanisms, they form a powerful defense system that helps us to remain safe from infections. So there was neutralizing, opsonization, and the complement activation mechanism. Those three. are very important in the formation of a very powerful defense system. We've seen how the antibodies function, but now what or how the antibodies work. But what are the functions? What are the functions? Okay, what are the key functions of the antibodies? So one is neutralization, we saw that. The second is opsonization, we saw that. The third is complement activation, we saw that. Now the fourth is agglutination. Agglutination is clumping together, kufanya vitu ishikane. So antibodies can bind to multiple pathogens at the same time. The antibody at that particular point is acting like a glue. So once it binds to the pathogens, those pathogens now clump together. Zinashikana. So zikishashikana, they are prevented from spreading. Okay? So once zimeshikana ziko pamoja, it's now easier for the immune cells to come and engulf, then remove those pathogens that have been put together using the antibody. So that process of putting them together, that process of making them to hold onto each other, to bind onto each other, that's what we are calling agglutination. The fifth function is the antibody-dependent cellular cytotoxicity. That is, the antibodies help in directing the killer cells. Okay? So some antibodies will help the natural killer cells to recognize a cell that is infected, a cell that is abnormal, so that those two cells can be destroyed. Okay? So some antibodies will help in the identification of infected and abnormal cells by the natural killer cells, so that the natural killer cells can now kill either the cell that is infected or the cell that is abnormal. Once that cell has been killed, then whatever was inside that cell will not spread. Okay. The mucosal immunity. Certain antibodies, for example, the immunoglobulin A, is found in tears, in mucus, in saliva. So such antibodies that are found in the mucosal lining, they help to prevent pathogens from entering the body through the mucosal surface. Okay. The passive immunity. Okay, so antibodies also help in the development of passive immunity, or the transferring of infection, of protection. So some antibodies can be passed from one person to another, so that they help in the provision of temporary immunity. We saw the example where some antibodies, for example, the immunoglobulin G, can cross the placenta so that it can protect the newborns. Also, through breastfeeding, we have immunoglobulin A, which can help to protect the infants from infection. This is passive immunity because the baby is receiving antibodies that are already made. Okay, so that is passive immunity. What about immunological memory and the concept of vaccination? So as we've seen, our immune system is designed to remember some of these past infections. That aspect, that ability of the immune system to remember, is what helps the immune system to fight some of these pathogens. Okay? So if it can remember a pathogen, then the same pathogen attacks you, your immune system is able to help you fight it off before it causes illnesses. That is what we call immunologic memory. So when we first encounter a virus or a bacterium, our immune system takes time to recognize and attack it because it has never encountered it. It doesn't have antibodies against it. But after this first infection, special cells, the memory B cells, the memory T cells, remain in our body. The importance of these memory cells is that they will allow for a faster immune response if we ever encounter the same pathogen again. Okay. Then a stronger attack that clears the pathogen before it even causes illness. So the attack is now against the pathogen. Okay. So it is a faster and stronger attack against the pathogen. So this could explain, for example, why most people just get some diseases once. Then anytime you encounter the same disease-causing pathogen, your body uses the memory B cells and T cells to clear it. Okay? So that you don't become sick of the same pathogen again. So that now lays the basis on how vaccines can use immunological memory. Because now, ideally, vaccines are just taking advantage of the ability of our immune system to remember. Okay? So, if we can train our immune system in a safe and controlled way, then our immune system can keep remembering these pathogens. And we are likely to have protection for a long time. That protection is now vaccination. So instead of exposing the body to a disease-causing microorganism, vaccines will remove the harmful part of the organism and expose the harmless part to you. So what is harmless cannot cause disease, but it is able to elicit an immune response from your body. So either the virus has just been inactivated, the virus has been weakened, or at times they just get some proteins of the bacteria. Okay, then they can leave it to be exposed. For the COVID-19 vaccine, they used the messenger RNA technology so that they can create something that your body will respond to, where your body will create memory B cells about. So once the immune system encounters these harmless parts, it will produce antibodies and memory cells. The part that your body is being exposed to, the part of that disease-causing organism, is harmless in the case that it cannot cause disease. But it is still able to trigger your body to produce antibodies. A section of those antibodies will be the memory B cells and the memory T cells. Those are the ones that will stay, so that in case there is an infection by now the real disease-causing microorganism, then your body is now able to mount an immune response. Okay? So that will mean that if you are exposed to the actual pathogen later, your immune system will respond immediately and powerfully so that it can prevent infection. That is the whole concept of vaccination. Expose the body to something that is harmless, cannot cause disease. But whatever you're exposing the body to, as much as it cannot cause disease, let it be able to cause an immune response. The immune response will lead to the development of antibodies, the B cells and T cells. Okay? Some of them will be part of the memory B cells and memory T cells. So that in case in the future you encounter the real pathogen, then you can now mount a response immediately and powerfully. That is now what will prevent you from developing the illness. Okay? There are examples of immune memory here. Okay? But why vaccination is important? It protects individuals from severe illness and complications. It reduces the spread of infections in the community. Okay? then it prevents dangerous outbreaks of diseases. Things like measles, polio, we've been able to control them just because of vaccination. So, due to vaccinations, there are so many deadly diseases which once killed people in millions, like smallpox, that have been either controlled, if not then totally eradicated from the world. Okay? And it is therefore corrective, we say, that among the most powerful tools in modern medicine are vaccines. You remember even during the time of COVID, what was able to contain COVID largely were vaccines. When the vaccination was rolled out, then the control of COVID became more of a reality. Okay. So among the most powerful tools in modern medicine are vaccines. How vaccines work is adaptive immunity, where your body is exposed to a harmless version of the disease-causing pathogen. That is harmless in a way that it cannot cause disease, but it is still able to trigger an immune response. Upon triggering the immune response, it leads to the development of B cells and T cells, which are kept as memory cells. The memory cells are now what leads to the immunologic memory. So in case you are exposed to that pathogen, then these T cells and B cells, which were kept in memory, will be called upon to mount a rapid and strong attack to the disease-causing pathogen. In that case, then, that disease-causing pathogen will be eliminated before it actually causes illnesses. Okay? Yeah, so thank you very much. That is about adaptive immunity. I hope I've explained it, but read the notes, listen to the video. Should you have a question, reach out to me through the email, which is down here, or when we meet in class next. Asanteni sana. I'll do a video next about B cells, okay, and antibodies. I'll do a video next about them. Thank you very much.